Welcome back friends. Welcome back in another video tutorial for microbiology with Shagnik. So today we will going to start another video lecture series where we will going to talk about the practicals. The practicals related to our field. So today in the first video of this playlist we will going to talk about a difference between positive staining and negative staining. We all know how much important staining techniques are, right? Because bacteria are semi-transparent in nature. That is why we cannot visualize them until we color them, right? Although staining has several drawbacks, for example, as staining is a chemical agent, that is why we cannot see bacteria living, right, when we stain them, right? That means with the help of staining technique we usually visualize dead bacteria, although we have now various type of other microscopical techniques where we are not using any kind of stain, but still we can visualize them, for example phase contrast microscopy, right? So today's video is all about staining. So we will going to do a basic difference between positive staining and negative staining. So positive staining is the general type of staining technique that we usually use to perform in our microbiology lab or any kind of biological lab. And negative staining is a modification of positive staining where we used to stain those type of bacteria that cannot be stained with the help of positive staining. Okay, so first clear one thing, that bacterial surface is negative in charge. Okay, so if we want to stain them, so we have to use some type of agent which should be positive in charge. That's why they will adhere with this bacterial surface, right? So that is why the name positive staining, because here we are using positively charged basic dye, for example crystal violet and saffron, that will color the bacteria. Okay, on the other hand, negative staining, the main goal of negative staining is not to stain the bacteria, instead to stain the background. Okay, that means if I show you the picture of negative staining, then you can understand. This is the picture of negative staining where you can see that bacteria are clearly visualized in white against the dark background, right? So here our main goal is to stain the background. So now try to understand, as I have told you that bacterial surface is negatively in charge. So if we going to add a stain which is negative in charge, then bacterial surface will going to repel it, right? And as a result only background will be stained, not the bacteria. So negatively charged acidic dye, for example nigrosin or Indian ink, will going to act as a negative staining technique. Okay, so why we are using negative staining? As I have told you, the bacteria which cannot be stained in positive staining can be stained with the help of negative staining. For example, bacteria those have a lot amount of polysaccharide in their surface can be stained by negative staining, because remember in negative staining the washing process is not too much required, and this is very useful to stain bacteria having polysaccharide in their surfaces. Again, capsulated bacteria are also can be stained with the negative staining. Okay, again in the positive staining technique heat fixation is very important, but in negative staining we don't require. On the other hand, in negative staining there is no requirement of heat fixation. Now let's try to think about this. Those bacteria who may be distorted due to effect of heat cannot be well stained in case of positive staining, but can be stained well in negative staining, right? So that is the thing. So that's how, depending upon their composition, mainly chemical composition, we can use negative staining to stain and visualize them well. Okay, so now in case of positive staining, what are the steps? First we have to prepare the smear. If you are using liquid culture, then okay, you will take a loop full of culture, you will make the smear, you will first air dry it, then heat fix, right? After that you will add the stain, right? After 1 minute you will rinse the slide with gentle tap water, and then you can examine it with help of any kind of student microscope. But in case of negative staining, the process is quite different, which I will going to show you with the help of this picture. So two kind of processes available as the picture depicts here. First nigrosin, which is the stain, is taken at one termini of the slide. Then they have taken the inoculum, right, and made the smear by tilting the slide at a 45-degree angle. Okay, once, and here they have used a simply another slide, right, to at a 45-degree angle to make the smear, right. Now after preparation of the smear, they have air dried it and observed it. You may also add the inoculum first, then you may add the nigrosin, and then you use another slide at a 45-degree angle to make the smear. Both are possible. Okay, so that is the idea of negative staining. That means they are doing what? They are taking the culture at one termini of the slide, right? Then they have added the nigrosin or Indian ink, what you can say. After that, they have used another slide at a 45-degree angle to make the smear. Okay, and after that they have air dried it and then they have observed. So that is the idea of negative staining. Okay, and remember it very carefully that in negative staining heat fixation is not present, as well as the washing step is not required. That is why the bacteria which are capsulated as well as bacteria who are rich in polysaccharide can be well stained with the help of negative staining. Okay, and the stains which are used in negative staining are acidic dye, right, and negatively charged, for example nigrosin, Indian ink. So that's the video for today's class. So I hope this video will be helpful for you. So thank you for listening to this class.